Well, my dear students, we are now studying the chapter respiration. The respiration is a very important physiological phenomenon in organisms, maybe plants or animals. But the thing is, when the respiration is studied, we have studied that there are two types of respirations in the yesterday's class. One is aerobic respiration and another one is anaerobic respiration. Aerobic respiration takes place in two steps or three steps. First one is glycolysis. Second one is what is called the ETS cycle. And yesterday we have completed glycolysis and we have seen how glucose is converted into pyruvic acid. And during the conversion of during the complete process of glycolysis, what all are produced in the process? That is 2 molecules of pyruvic acids, 2 molecules of NADH2, highly reducing agent, and 4 molecules of ATP. Total ATP produced in glycolysis are 10, but 2 ATPs we have invested in the beginning of the glycolysis. So total profit of ATP in the glycolysis is totally 8 ATP. Okay, now let us see the second step. Okay, after the glucose when entered into cytoplasm, in the cytoplasm glycolysis takes place. So the site of glycolysis in the cell is the cytoplasm, please remember. So when the pyruvic acid is produced by the glucose during the process called as glycolysis, pyruvic acid is now converted into another compound. Say this is called as Krebs cycle. Krebs cycle. Okay. So what is Krebs cycle? So here we can see here pyruvic acid. Say it is a 3-carbon compound, and pyruvic acid is converted into one compound here, what is known as acetyl-CoA. I am writing here. Acetyl-CoA means it is acetyl coenzyme. So this acetyl coenzyme is produced now here, and during this, one molecule of carbon dioxide is released. Okay, one molecule of carbon dioxide is released, and one molecule of, so many things are happening here. NAD are taken, one molecule of NADH2 are released. NAD is taken, NADH2 are released, and one molecule of, so you can write here, coenzyme A is taken and coenzyme A is released once again. Coenzyme A is released. Okay, coenzyme is taken here, just not released here. It is coenzyme is entering in the process. So this is actually happening here. So this is called as conversion of pyruvic acid into acetyl coenzyme A. Acetyl coenzyme A. I will just repeat once again. One molecule of pyruvic acid is converted into acetyl coenzyme A, during which one molecule of NADH2 is liberated, one molecule of carbon dioxide is liberated, and one molecule of coenzyme A is taken inside. So all these things are making here, and acetyl coenzyme A is containing 2 carbons. Please remember, pyruvic acid is a triose, 3-carbon compound. One carbon dioxide has gone here, that means one carbon has been removed. So acetyl coenzyme A is now having 2 carbons. It is having only 2 carbons, acetyl coenzyme A. Okay. Acetyl coenzyme A now, it is now present in the cytoplasm. Please remember, it is now still present in the cytoplasm, and acetyl coenzyme A is now entering into mitochondria. Acetyl coenzyme A is now entering into mitochondria, and it is going inside the mitochondria, and in the mitochondrial matrix there is one compound called as OAA. I am writing short forms here. OAA. So OAA it is oxaloacetic acid, OAA. It is a 4-carbon compound. 4-carbon compound. And acetyl coenzyme A is now, so when acetyl coenzyme A enters into mitochondrial matrix, first it combines with acetyl coenzyme A, and acetyl coenzyme A reacts with oxaloacetic acid and produce one important thing, citric acid. It is producing citric acid, and here one molecule of coenzyme A is liberated. One molecule of coenzyme A is liberated, and the enzyme required here is citrate, very important, synthetase. The enzyme which is responsible for the conversion of, for the reaction between acetyl coenzyme A and oxaloacetic acid to produce citric acid is the citrate synthetase. And this is having 5 carbons. This, you must be very careful here. 4 carbons and this is 6 carbons. Please remember, this is 6-carbon compound, hexose molecule. 6-carbon compound, because 2 carbons from acetyl coenzyme A, 4 carbons from oxaloacetic acid, it is producing 6-carbon compounds. And the citric acid is now converted into one more compound here, that is cis-aconitic acid. Cis-aconitic acid. Cis-aconitic acid is produced. Cis-aconitic acid is again 6-carbon compound. 6-aconic acid, and during which one molecule of water is liberated. Dehydration. And the enzyme responsible for this is aconitase. Aconitase. This is called as aconitase. So aconitase enzyme converts citric acid into cis-aconic acid, and this cis-aconic acid is now converted into cis-aconic acid is produced, and cis-aconic acid is now converted into isocitric acid. Isocitric acid. So this is also a 6-carbon compound, and during which one molecule of water is taken inside. Water is taken inside. See here, fun. One molecule of water is taken. Here one molecule is liberated. Here one molecule is taken. Here one molecule is taken. So cis-aconic acid, and it is converted into isocitric acid. Isocitric acid is converted into oxalosuccinic acid. Oxalosuccinic acid. This is also a 6-carbon compound. Oxalosuccinic acid. Oxalosuccinic acid, and oxalosuccinic acid is produced, and the enzyme responsible for is succinase. Okay, succinase. Oxalosuccinic acid is now converted into one more compound called as alpha ketoglutaric acid. Alpha ketoglutaric acid. Alpha ketoglutaric acid is produced here, but here one important thing is happening. One molecule of carbon dioxide is liberated, please remember, and one molecule of NAD is taken, one molecule of NADH2 is liberated. One molecule of NADH2 is liberated. Now, NADH2 is liberated, and the alpha ketoglutaric carbon content is 5 carbons. Please remember, oxalosuccinic acid is having 6 carbons, 1 carbon dioxide has gone, and because of this alpha ketoglutaric acid is containing 5 carbons. And alpha ketoglutaric acid is then converted into one more compound, what is known as succinyl coenzyme A. Succinyl coenzyme A. Here one molecule of carbon dioxide is liberated once again. and NAD is taken, NADH2 is liberated. Okay, and so here the enzyme that required is oxalo, or simply you can write decarboxylase. That is the enzyme which is required. You can write oxalosuccinic decarboxylase, you can write, no problem in that. Or dehydrogenase, you can write. Both reactions are taking place here. And the enzyme responsible for the conversion of alpha-glutaric acid into succinic acid, the enzyme responsible for this is alpha-ketoglutarate dehydrogenase. So this is the enzyme responsible, because from alpha-glutaric acid one molecule of hydrogen is removed. So because of that the alpha-glutaric dehydrogenase is produced. And succinic acid is then converted into one more compound, what is known as succinic acid. And succinyl coenzyme A, the carbon content is 4 carbons, because one more carbon dioxide has gone now. It is only 4 carbon. This is also 4 carbon. But here one molecule of GDP is taken, guanosine diphosphate. It is equivalent to ATP only. GDP is taken, one molecule of GTP is liberated. Okay, GTP is liberated, and the enzyme responsible for this is kinase. I have told you already, wherever ATP is involved, you can write it as kinase enzyme. And succinic acid is converted into one more compound called as fumaric acid. And during this fumaric acid, one more important thing is happening here. FAD is taken, flavin adenine dinucleotide, and FADH2 is liberated. High reducing power, and here you can write dehydrogenase enzyme. So what dehydrogenase? Succinic dehydrogenase you can write, or simply you can write dehydrogenase enzyme. And fumaric acid is converted into one more compound called as malic acid. Malic acid is produced, and malic acid is produced, and during which one molecule of water is taken inside. Okay, malic acid is produced, and the enzyme responsible for this is succinate, or dehydrogenase. Once again, dehydrogenase enzyme. And malic acid is then converted into oxaloacetic acid, and during the conversion of oxaloacetic acid, so what happens is, during this one molecule of NAD is taken, one molecule of NADH2 is produced. One molecule of NADH2 is produced, and the enzyme responsible for this is malate dehydrogenase. So this is the enzyme responsible for the conversion of malic acid into oxalic. See, when the acetyl coenzyme has come inside the Krebs cycle, inside the mitochondria, we have taken OAA, oxaloacetic acid. After this entire cycle, what is the aim? Returning the oxaloacetic acid safely to the mitochondrial matrix, because the oxaloacetic acid is required for the next Krebs cycle. Whatever you have taken, if you consume everything, so the purpose will not serve, because the oxaloacetic acid is required for the next cycle of Krebs cycle. So it has to be returned back, regeneration. So oxaloacetic acid is regenerated. So this is called as the Krebs cycle. But one thing we have to think here, why this is called as Krebs cycle? Because H.C. Krebs, the name of the scientist, who has isolated all these intermediate substances, and he was the first person to discover what is Krebs cycle. So in order to give respect to the scientist or the researcher, this particular cycle is named after the inventor Krebs. So it is called as Krebs cycle, and for this he was awarded with Nobel Prize in 1962. So this is the most important aspect. And some more names are there for this. First one is called as Krebs cycle, and second one is called as citric acid cycle. The second name for this cycle is citric acid cycle. Krebs cycle is also called as citric acid cycle because the first stable product produced in this cycle is citric acid. See here, oxaloacetic acid reaction. So there are different names for Krebs cycle. First one is called as Krebs cycle. Citric acid, because the first stable product produced is called as citric acid, so it is called as citric acid cycle. Another one is called as tricarboxylic acid cycle or TCA cycle, because tricarboxylic acids, all of you observe this, from this to this you can observe all are acids here. What is what you call the pyruvic acid, citric acid, isocitric acid, cis-saccharidic acid, oxalosuccinic acid, alpha-glutamic acid, succinic acid, fumaric acid, malic acid, and oxaloacetic acid. So all these acids, they contain carboxyl group, COOH group, and not only carboxyl group, they have 3 carboxyl groups, so they are called as tricarboxylic acid cycle. All these names are given for this. Not only this, we have to understand what is the energy outcome of this particular Krebs cycle. So here you can observe how many NADH2 are produced here. NADH2 NADH2, NADH2, and NADH2. 4 NADH2 are produced here in the Krebs cycle. But I am writing here 4 NADH2 and into 2. You can observe here 4 NADH2 into 2. What is this 2? So 2 means from one molecule of glucose, 2 pyruvic acid are produced. This entire thing has taken place for one pyruvic acid. Another pyruvic acid produced by the same glucose is waiting. So that also enters into the same pathway. So because of that, this is called as producing 2 ATP, 4 for one NADH2. 4 NADH2 are produced here, and 4 NADH2 are multiplied by 2 because for one molecule of glucose, 2 Krebs cycles are taking place. So one thing. Second thing is, from one NADH2, 3 ATP molecules are produced. So 3 into 2 into 4 is equal to 24 ATPs are produced here. So 24 ATPs are produced, and there is FADH2 also. See here, FADH2 is also produced. FADH2 1 into 2 because another cycle, and one FADH2 produces 2 ATP molecules. So 4 ATP molecules are produced from FADH2. From NADH2, 24 ATP. From FADH2, 4 ATP, and 1 GTP is produced here. See here, GTP. So this GTP is producing one GTP is produced here. GTP is equal to ATP. Guanosine triphosphate and adenosine triphosphate, only the nitrogen base is different. So GTP is equal to ATP. 1 ATP in one cycle, but 2 cycles are taking place. 2 ATPs are produced. From Krebs cycle, total 30 ATPs are synthesized. 30 ATPs are synthesized, and here one thing we have to observe, from starting to end of this particular cycle, have you invested any ATP? No ATPs are invested. So whatever you are getting, that is the profit only. But from glycolysis you are getting 8 ATP as a profit. 8 ATPs are produced here, and this 8 ATP is the profit. So you have to add 8 ATP to 30. Total 38 ATPs are produced in the Krebs cycle. So that is the total profit of ATP in the Krebs cycle is 38. Okay. if the question is asked, so you have to first understand how the question is asked. Pattern of question asked here you have to understand. So what you have to understand is, what is the total number of ATPs produced by one molecule of glucose in aerobic respiration? Aerobic respiration is the one which is producing 40 ATPs. Total number of ATPs produced. So style of asking question is different. So what is the total profit of ATP in aerobic respiration from one molecule of glucose? That is 38 ATP. Okay, another important thing comes here. Okay, NADH2 are produced in the Krebs cycle. One NADH2 is also produced in glycolysis. How this NADH2 produces 3 ATP? How the FADH2 is producing 2 ATP? Where it is producing? So one more step of aerobic respiration we have to study, what is known as ETS cycle, electron transport system cycle. So for that you need the idea of mitochondria. Mitochondria is double membrane structure, and this double membrane structure has got outer membrane. Outer membrane is here, and this is called as outer membrane, and this is called as inner membrane. And in outer membrane has got two parts here, outside of outer membrane, inside of inner membrane, outside of inner membrane and inside of inner membrane. Like this, there are two things which are very important here. So two things are present here. So in between this you are finding so much of the liquid matter, and this is called as matrix. That is called as matrix. In the mitochondrial matrix, the Krebs cycle takes place, and whatever the products are produced in Krebs cycle, they are now, products means NADH2 and FADH2, these two are now entering into the inner membrane, what is known as the crista. This is the enlarged crista here. The crista are having so many racket-like structures. These are called as F1 particles. These F1 particles are involved in ETS cycle, and this ETS cycle is taking place. ETS cycle is converting NADH2 into 3 ATP, FADH2 into 2 ATP. How it converts everything, let us see in the next class. Okay, and in this one, respiration means what we are always giving the definition. Oxygen is entering, carbon dioxide is coming from the process. Or simply we will tell inhaling of oxygen and giving out carbon dioxide. It is not the case. But you have seen aerobic respiration has got 3 steps. First step is glycolysis, second step is Krebs cycle, and third step is ETS cycle. You have already studied glycolysis. Have you seen the entry of oxygen anywhere in the glycolysis? No oxygen has taken part. Oxygen role has not been started in glycolysis. Okay. Have you seen the role of oxygen in the Krebs cycle? Already second step is over. No, oxygen is not at all involved here. So already 80% of the respiration is over. No oxygen has seen here. But the oxygen role is observed only in the last stage of respiration, that is ETS cycle. So when the oxygen enters, ETS cycle completes. Not only ETS cycle, the respiration process is completed. So that is why the entry of oxygen in the ETS cycle is termed as terminal oxidation. So addition of oxygen is at the end of the ETS cycle, so it is terminal. So in the terminal part of the ETS cycle, the oxygen enters, so it is always called as terminal oxidation. That is very important aspect to be remembered. So this is about this one. And one more thing you have to remember always, in the ETS cycle, in the aerobic respiration, from glycolysis, from Krebs cycle, and the products of Krebs cycle, they enter into ETS cycle, whatever it may be, total number of ATPs produced are 38 ATP. And these 38 ATPs are equivalent to what? They are equivalent to 686 kilocalories of energy. 686 kilocalories of energy are produced now. And if you 686 divided by 38, if you do, whatever comes here, that is the value of 1 ATP. 1 ATP molecule, that is, I think it is 3.48 something. So the value of 1 ATP, the energy content of 1 ATP is 3 point something. So 3 point something is the ATP value. So that you actually deduct this one, and so if you calculate, you will come to know. So this is what is known as the respiration. and this respiration I have completed, that is aerobic respiration. And in this aerobic respiration we have to understand what is aerobic respiration, what are the steps involved in aerobic respiration, what is glycolysis, what are the steps involved in glycolysis, what is Krebs cycle. So between Krebs cycle and glycolysis there is a connectivity, that is conversion of pyruvic acid into acetyl coenzyme A is the connecting link, because pyruvic acid will not directly enter into mitochondria. First pyruvic acid is converted into acetyl coenzyme A. Acetyl coenzyme A now enters into mitochondria. All these are very crucial points, very important points. You have to always remember, and you have to understand the things very critically, and you have to write. Okay, so this thing I have completed so far. In the next class, next video class, I am going to deal with the most important part, what is known as the ETS cycle. And after the ETS cycle, I will be dealing with anaerobic respiration. What is anaerobic respiration? How many types of anaerobic respirations are there? What are the alternate names for that? Everything. I think already I have prepared the notes, and I will upload the notes tomorrow, and I will also complete anaerobic respiration in detail, and also the RQ, respiratory quotient, it may be asked in the examination, that also I am going to deal tomorrow, and factors affecting respiration, and industrial use of aerobic respiration. All these things I am going to continue tomorrow. So if you have any doubts, any queries, so you can ask me anytime. So you just feel free to call me, and already I have given my mobile number to you all of you know already. So you just contact me and get your difficulties clarified, because respiration and Krebs cycle in the aerobic respiration is very complicated. So if you understand once very clearly, you will not forget. But understanding critically, scientifically, and verbally, that is very, very important. You have to understand, and why this is called as this one, why this is called as this one, why this is called as Krebs cycle only, why this is called as citric acid cycle. So all these are very critical. You have to understand, and many things what I am explaining here, you will not get those points in any of the books. Because of my experience, I am telling. So you have to first understand what is happening here. So then in the next class, I am going to deal with all these things. So do well and take care of your health, and study well. So goodbye.